Good afternoon, dear colleagues. It is my pleasure and privilege to welcome you to this symposium on milk bioactives and probiotics in children, current trends and future directions by Nestle Nutrition Institute. I am Dr. Randy Ortula. I am the chairperson of the symposium and I will be the moderator as well. We have two topics for this afternoon, prebiotics and probiotics in children, current trends and future directions, which I will lecture after. And the second topic on functional insights in HMOs, key early life bioactives, will be delivered by Dr. Hannah Tigler. After these two topics, there will be an open forum wherein you can type in your questions. Our key messages for this afternoon would be for you to review the importance of age-appropriate maturation of the gut microbiome and its impact on the immune system, as well as understand the impact of research and science on milk additives and probiotics on clinical practice. And lastly, we get updated on the latest research on microbiomes and HMOs. I am a professor in pediatrics as well as a pediatric gastroenterologist with more than 30 years of teaching experience as a professor at the College of Medicine at Far Eastern University Medical Center and the New Era College of Medicine. My interest in nutrition brought me to work as a member of the Committee on Philippine Dietary Reference Intakes in 2015 with the Food and Nutrition Research Institute of the Department of Science and Technology in the Philippines. And aside from my teaching positions, I am the former head of the section of pediatric gastroenterology, pathology, and nutrition, and is now the head of the Medical Internal Systems Specialty Division and the Pediatric Transplantation Technical Working Group at the Philippine Children's Medical Center. And these are my disclosures. I am an advisory board member or even a speaker for Nestle Nutrition, Nestle Nutrition Institute, Nestle Health Science, and Wyatt Nutrition. Good afternoon and welcome to this lecture on prebiotics and probiotics in children, current trends and future directions. The recent years saw an exponential rise in the number of researches on pre and probiotics as their ability to modulate certain health conditions in pediatrics become known. Research on the human microbiome offered new insights on their role in regulating body physiology, host protection, and modulation of immunity. The cumulative numbers of studies show thousands of them directed towards elucidating the functions and effect of these bioactive components. If you look at the clinicaltrials.gov database, by March 2019, there are around 1,341 studies. And add to this the World Health Organization International Clinical Trials registration platform of another 278 studies, and would add to about 1,619 studies for that month alone. And it's interesting to note that the most studied probiotic strains were actually Lactobacillus rhamnosus GG or LGG and Bifidobacterium animalis species lactis or BB12. Now, for us, the key for the efficacy of probiotics to certain conditions lie actually in the understanding of certain key concepts. For example, we must understand that the effects of these probiotics are strain-specific and has been dose-tested in studies. Let's also understand that each of these probiotics have their own mechanistic action, and thus not all probiotics are created equal, and therefore they don't have also equal efficacy. But also we must understand that it is still scientifically sound to consider certain individual effects of these probiotics when they are applied to common conditions that can result to similar outcomes. Here are the following applications of probiotics in pediatrics with their strains that have been recommended. And the best documented efficacy for certain probiotics is actually on the treatment of infectious gastroenteritis, prevention of antibiotic-associated diarrhea, and nosocomial diarrhea. But for H. pylori infection, there's still no clear recommendations what strain to use because of the limited evidence in pediatrics. Also, for the allergic diseases like your atopic dermatitis, allergic rhinitis, and asthma, No firm recommendation of strains to use were made because there's little or no difference in improving the quality of life in patients who are suffering from eczema symptoms. And thus, the World Allergy Organization suggests that you only use probiotics when you are in a high-risk groups. For the functional gastrointestinal disorders, like functional constipation, functional abdominal pain, and irritable bowel syndrome, there is little evidence and no firm conclusions that can be given for the use in these conditions. However, for infant colic, the use of Lactobacillus reuteri DSM 17938 has been made particularly for the breastfed infants. This is so because although the probiotics are promising treatment options for certain functional gastrointestinal illnesses subtypes, the overall quality and quantity of the evidence are relatively weak. With the results of the previous meta-analysis, several regional recommendations for the use of probiotics in children has been made. There were recommendations coming from the Asia-Pacific region, from the Southeast Asian region, Europe, Latin America, and the United States. But with the recent review by DePorter and Van den Pla for the pediatric conditions wherein probiotics were studied, let me direct your kind attention to the recommendations. For the functional gastrointestinal disorders, your probiotics are not recommended for irritable bowel syndrome, constipation, and regurgitation. But however, for infantile colic, there has been some recommendation of using Lactobacillus reuteri DSM 17938, but mainly for the breastfed infants. There are no recommendations also for atopic dermatitis, asthma, and allergic rhinitis. But recommendations have been made particularly for antibiotic-associated diarrhea and its prevention, Clostridium difficile-associated diarrhea, and as well as your nosocomial diarrhea prevention. But for acute gastroenteritis, either treatment or prevention, the latest reviews show that there might not be an effect at all, and thus the jury is still out there debating whether to use what strain or if ever to use probiotics at all for these conditions. Now, prebiotics like your human milk oligosaccharides are very important also, and majority of your HMOs are metabolized by your intestinal microbiota and excreted only in the urine and feces, and only about a small percent of them are actually absorbed in the circulation. Now, the role of your milk oligosaccharides are important because from studies, they have shown beneficial roles in the infant's digestive tract. And this can be exerted either through a direct or indirect interaction with the gut microbiota. Researchers have shown that they can support the beneficial bacterial growth as well as its antipathogenic effects, and they can modulate intestinal epithelial cell response. Now, the possible therapeutic applications, therefore, of your human milk oligosaccharides has been made. There have been studies showing that it has therapeutic potential in food allergies. They have also positive effects against necrotizing enterocolitis, and as an anti-infective agent like antibacterial, antiviral, or even antifungal and antiprotozoan. And this would probably more dealt in a detailed manner later in the next topic. There have been studies also that your human milk oligosaccharides can actually do have an effect on the non-communicable diseases like obesity, type 1 and 2 diabetes, and cardiovascular disease. Animal studies already been showing that there's improved cognitive ability, learning, and memory. So therefore, the addition of your HMOs in infant formulas may actually be an important therapeutic strategy to target certain windows of opportunity among the non-breastfed infants. However, we must note that majority of our conclusions may be inconclusive and even with conflicting results. And why is this so? Because there are several reasons. One is that clinical trials were conducted in different ways. The trials for these ones have not been standardized because of the difference in the strain formulation, combination, and even the dosing. There's also heterogeneity high risk of bias, and small sample size in the design of some of the studies. The studies with different patient populations, different reported endpoints and outcomes were grouped together inappropriately in some. There's also a marked regional variation in the distribution of certain enteric pathogens that can be affected due to the difference in socioeconomic and nutrition status, the diet, the culture, and even genetics that can now diversify the outcome of these probiotics. Lastly, prebiotics and probiotics have mostly been studied alone, whereas we know that they exist in a diverse pool within the breast milk itself and interacts with the gut with diverse actions. Now, actually, we might be losing sight that breast milk, which contains both prebiotics and probiotics, aside from its many diverse bioactive compounds, should actually be viewed and studied as a complex biologic system that can interact both with external and internal factors to influence now the growth and development of infant. Now, the common approach of analyzing a single component of breast milk may now be insufficient to understand its role that contributes to the matrix of the components that are present and that can now influence both maternal and child health. So therefore, there's actually a need for us to shift how we look at breast milk, looking it as a whole and not the sum of its parts to reveal the multifunctionality and interaction of its component that drives growth and development in early life. So the challenges for future research remains, and these are the following. Further research should be undertaken whether your HMOs or probiotics may also have a therapeutic rather than just a protective effect in the infant gut. We need more cutting-edge research using state-of-the-art nutritional analytics to improve our understanding of the complex biological interactions following supplementation with probiotics and some of the novel bioactive ingredients. More researches should be done looking at the cost-benefit of treatment of illnesses that can affect children. We also have to do more well-designed researches involving population of high-risk children from the underdeveloped and the developing countries, particularly on certain health conditions with high morbidity and mortality. We also need large community researches that would provide convincing data that are powerful enough for these clinical interventions to be included in public health policies to benefit more of the population. So in summary of this short lecture, the data suggests that the gut microbiome does have an impact on the different health conditions in infants and children. However, the clinical outcomes from studies are often contradictory and even inconclusive. There is a great need for more robust, large-scale trials that demonstrate the interactions of these bioactive components that would provide convincing data so that their use can be translated to public health policies to benefit more of the population, especially in the developing countries. Now, over the past few years, researches have been undertaken to try to improve not only the macro and the micronutrient content of infant formulas to make it closer to the composition of breast milk, but they have also included now additional bioactive components with potential health benefits for infants. Further large-scale, well-designed studies are needed to demonstrate if these novel bioactive ingredients do have the same functional effects and health impact as components when they are found in human milk. So with that, I thank you very much for listening to this lecture. Our next speaker would be Dr. Hannah Tigler, who is a microbial glycobiologist and focuses on elucidating the role of glycans in microbiota host interactions. After earning her PhD in bioscience engineering from the University of Leuven in Belgium, she worked as a postdoctoral researcher at the University of Wageningen in the Netherlands and ETH Zurich in Switzerland. Her current role at Nestlé Research Switzerland leverages her expertise to understand how dietary glycans affect development and help for nutritional application. So with that short introduction, let's proceed with the lectures.
Hello, everyone. Today, I will tell you something more about functional insights in human milk oligosaccharides, or HMOs. These are key early life bioactives. I'm Hannah Tigler, and I work at Nestlé Research in the Institute of Health Sciences in Lausanne, Switzerland. Let's dive in. Early life offers a critical window to lifelong health because it's in this time period that the microbiome and the immune system are maturated. Indeed, when an infant is born, its gastrointestinal microbiome is sterile. It means that during the birth, it receives the first microbes from its mom, mainly Enterobacteriaceae. Over the next few weeks and months, the gut becomes increasingly anaerobic, and we see that more species start to pop up. These are mainly bifidobacteria, which is largely steered also via breastfeeding. This bifidobacterial dominance is really key to early life and is a typical feature of an early life microbiome. Upon the introduction of more diverse complementary foods, so diversification of the diet, we also see a rapid diversification of the microbiome. We see more adult-type species popping up, which further a trend that is further established during the following months and years, until a mature microbiome is established, which is highly diverse and resilient. This will offer a strong basis for lifelong health and help the infant to combat infections and to recover from dysbiotic events. The increase in microbiome diversity over the course of early life goes hand in hand with also the training of the immune system. The immune system really is trained during this critical window, where it learns to distinguish between good bacteria and bad bacteria. It learns to distinguish pathogens and to then react to prevent infections. So microbiome maturation goes hand in hand with the maturation of the immune system. And beyond that, a healthy microbiome is also important to strengthen gut barrier function, also has a role in modulating the developing nervous system. You might have heard about the gut-brain axis, where we see that there is a link between the microbiome and the brain, affecting, for instance, mood or some disorders affecting the brain, and so on and so on. Evidence on this is still further developing. What we also see is that there's a strong link with the developing immune system, where the microbiome helps to educate it, as I mentioned before. Also, a good resilient microbiome helps in resisting colonization from pathogens. This means that pathogens have a harder time to establish themselves in a resilient microbiome. So really the gut microbiome development and in a strong way is linked to long-term health. But this early life is a critical window, but it's also a very cumbersome time because various factors affect this gut microbiome development. There are many risk factors that threaten the establishment of a healthy microbiome. For instance, the gestational age, the mode of delivery, the feeding mode, and antibiotic use might all influence gut microbiome development in early life. To give an example, we see, for instance, from research that C-section delivery and antibiotic use in early life affect gut microbiome development. When we see here, for instance, in gray here, we see this typical bifidobacterial peak in relative abundance in early life. We see that this really up till 3 months, which is typically around the time where complementary foods are often introduced, that the bifidobacteria dominate the microbiome of the infant. Then we see a rapid decline upon the introduction of complementary foods. All normal events. However, when we see that the infant is born via C-section, here depicted in red, we see that there is a delay in bifidobacterial dominance, and also that the relative abundance of the bifidobacteria does not reach the same height as in a normal trajectory. When we look at the trajectory for infants receiving antibiotics in early life, we see even a worse trajectory where often the bifidobacterial dominance is completely obliterated and not happening anymore. This has really strong effects on health conditions in later life. For instance, in infants delivered via C-section or receiving antibiotics, we see an increased incidence in lower respiratory tract infections, a larger incidence of allergic outcomes, like for instance, asthma, food allergies, and allergic rhinitis, and a higher chance to develop overweight and obesity in later life. Many of these risk factors, for instance, C-section delivery and antibiotic use, come together when an infant is born prematurely. Another factor strongly influencing microbiome development is diet. We know that breastfeeding stimulates gut microbiome development. Because here depicted in pink, we see that the alpha diversity of breastfed infants gradually increases over time, over the months. When we look at the abundance of bifidobacteria, then we see that over the first 3 months, there's a strong increase in bifidobacterial relative abundance, which is then goes down after introduction of complementary foods. In blue, we see the trajectory for an infant fed formula based on cow milk. There we see that there is a very rapid increase of the alpha diversity of the microbiome. We also see when we look at the bifidobacteria that there is not this typical peak in relative abundance of the bifidobacteria compared to other species, which is in line with the rapid diversification of the microbiota. So we see a very rapid introduction of several species and not this gradual increase like we see in breastfeeding. So we see that breastfeeding, or feeding mode in general, really strongly drives microbiome maturation. When we then look at breastfeeding, which is the golden standard for infant nutrition, we see that this comes from the fact that it not only has all the feed to feed the infant, but also its microbiome. Breast milk is full of nutritive components like lactose, proteins, and fat that are digested and have a nutritive value to the infant. These support healthy growth and development. But there are also many other bioactive components in breast milk. For instance, cytokines, antimicrobial proteins, antigens, and so on. Key of these bioactive components are HMOs, or human milk oligosaccharides. These are non-digested, don't really have a nutritive value to the infant, but they support the microbiome and immune function. They will support immunity, gut health, brain development, and learning. So they're really key to the infant's development. Looking further at these human milk oligosaccharides, we see that they're the third largest solid component in mother's milk. They range up to 5 to 15 grams per liter. Over 160 different structures are described, with an estimated 20 of them that represent over 80% of the total HMO mass. Most common HMOs found in mother's milk are not, or only to a minor extent, present in farmed animal milk. They can be subdivided in 3 groups. All are basically elongations of the milk sugar lactose, and if this lactose is elongated by a fucose moiety, we have the class of the neutral fucosylated HMOs, like for instance 3-fucosylactose, 2-FL, LDFT, and so on. Or the second class are the acidic ones, where acylic acid is added to the lactose, resulting in 3-silactose, 6-SL, and so on. Or more neutral moieties are added, like for instance, glucose, galactose, glucnac. And then we have these LNT, LNNT. These can be then further elongated to form more complex HMOs of the different classes. There's not only a strong structural diversity of the HMOs, there are also a large functional aspects that are linked to HMOs. HMOs are found to support microbiome development, as I showed you before, where they can have a kind of a prebiotic effect, but they also can have a direct antimicrobial effect by, for instance, preventing the colonization by different pathogens. They strengthen the gut barrier function, modulate the developing nervous system, and have an influence on cognitive development, and they also play a role in immune competence development. Taking now a look at our clinical understanding of these functionalities, we can benefit from the fact that HMOs, since recent, also can be produced in a technical setting. Technical advances have allowed for the industrial production of HMOs, which allows us to evaluate their functional aspects in randomized controlled trials. First, we evaluated 2-fucosyl-lactose, a neutral fucosylated HMO, which was then later complemented with a neutral non-fucosylated HMO, leading to 2 HMOs. And currently, we are running a trial in which we evaluate functional aspects of 5 HMOs, where all 3 structural main groups of HMOs are represented. But let's start at the beginning. What we learned from a clinical trial where infants received formula supplemented with 2-fucosyl-lactose, we saw that this resulted in a lower relative risk of an infant to have infections compared to infants receiving a control without a control formula that had no HMO supplemented. This effect was sustained in an endeavor where we looked at 2 HMOs. Infants receiving a formula supplemented with 2 HMOs, 2FL and LNNT, we had lower effect of upper respiratory tract episodes, and also they had a low risk to have these respiratory tract infections when they received the infant formula supplemented with the 2 HMOs compared to control formula. Looking at other effects of this 2 HMO formula showed that, for instance, infants receiving 2 HMO supplemented formula had a lower risk to require antipyretics during their first months of life, and also that this reduced the risk to require antibiotics during their first year of life. Also, during their first year of life, the infants receiving infant formula supplemented with 2 HMOs had a lower risk to experience lower respiratory tract infections, as depicted in the 2 graphs here below. Looking more in what could explain this, we looked at the Bifidobacterial abundance. As I showed you before, this really is important in early life, that there's a peak in bifidobacterial relative abundance during the first 3 months of age. Here we see indeed that in the breastfeeding arm, that there is a strong dominance of bifidobacteria, irrespective if the infant was born via cesarean or vaginally born. In infants receiving a control formula, not supplemented with HMOs, we saw that this bifidobacterial relative abundance was lower in cesarean-born infants. What then we saw in infants receiving the formula supplemented with 2 HMOs is that this 2 HMO supplementation could increase the bifidobacterial relative abundance so that it could increase this, bypass this effect of C-section on this bifidobacterial abundance in early life. Also, in vaginally born infants, there was an effect on a relative abundance of bifidobacteria in the test formula supplemented with the 2 HMOs. When looking at the microbiota composition in general, we see that in breastfeeding infants, fecal community type B is the most abundant one, here depicted in red. Compared to infants receiving formula without HMOs, the control group, we saw that their fecal community type C was the most abundant. Now, in infants receiving formula supplemented with 2 HMOs, we saw a strong increase in fecal community type B, showing that the microbiota community of these infants moved closer to the one of breastfed infants. When comparing the antibiotic use per fecal community type, we saw that infants falling into this fecal community type B had a lower chance of requiring antibiotics over their first year of life, compared to infants having a fecal community type C. In conclusion, this shows that the HMOs had a bigger effect in the case of dysbiosis, for instance, with the C-section-born infants. Moreover, HMO feeding drives the microbiome towards that of breastfed infants. And this microbiota community shift in the infant's fed formula with 2 HMOs relates to the antibiotic use over their first year of life. Currently, we are evaluating a randomized clinical controlled trial in which we evaluate the effects of an infant formula supplemented with not 2, but 5 HMOs. Here, also acidic silylated HMOs are supplemented, so covering all 3 big groups of HMOs. The purpose of this trial is to evaluate the tolerance and safety of this 5 HMO supplemented formula, as well as its effect on growth and the effect on gut health and immunity markers. The trial was set up as a multi-center, randomized, controlled, double-blind study of healthy term infants in Hungary, Bulgaria, and Poland. The trial also consists out of a non-randomized breastfed reference group. Infants receiving formula either received control formula, which consists out of intact protein consisting of 70% whey, 30% casein ratio, and 100% lactose, which is replaced in the test groups by either 1.5 gram per liter of the, or 5 HMO plants or 2.5 gram per liter of the 5 HMO plants, meaning that this is test group 1 and test group 2. So there are 4 arms in this study. Looking at the effect on growth, we saw that 5 HMO in a supplemented formula supports age-appropriate growth. The growth in these groups was non-inferior, and the weight for age and length for age z-scores were all similar between both test groups and the control, and tracked closely to the median of WHO growth standards. Moreover, the infant formula was well tolerated and supported a soft stooling pattern. When looking at the microbiota composition, we saw that at baseline, the breastfeeding group differentiates from the 3 formula-fed groups, but that over the time and at 3 months of age, we see that the both groups that received infant formula supplemented with 5 HMOs moved closer to the breastfeeding arm in a means of better diversity of their microbiome compared to the control group that did not receive 5 HMOs in their formula. This was also reflected in the abundance of bifidobacteria. Bifidobacterial relative abundance of especially test group 1 approached more breastfeeding abundance. When looking at a specific species of Bifidobacteria, Bifidobacterium longum subspecies infantis, which is a known HMO metabolizing strain, we saw that the relative abundance of this strain increased and moved closer to breastfeeding compared to the control group. Also, the relative abundance of other taxa was significantly changed in the test groups compared to control, and all moved closer to breastfed infants. When looking at a pathogen, Clostridioides difficile, which is known to cause diarrhea in infants and children, we saw that the relative abundance and prevalence of toxigenic C. diff was lower in the test groups compared to the control. It moved closer to breastfeeding, and while in breastfeeding, both in relative abundance and prevalence, the C. diff, the toxigenic C. diff, was non-detectable. Between the test groups, there was no significant difference between both the prevalence and relative abundance of this enterotoxigenic C. diff. Also, in manners of gut ecology, we saw that the infant formula supplemented with the 5-HMO moved the gut ecology closer to the one of breastfeeding, of the breastfeeding arm, where we saw an increase in acetate, a decrease in butyrate, an increase in lactate, and a decrease in the fecal pH. All markers showing that gut ecology moved towards the one of breastfed infants. Also, it was found that 5-HMOs support gut functional maturation. There, two markers are of importance. One is gut immunity marker SIgA. SIgA is an important gut gatekeeping molecule. It protects against toxins, viruses, and pathogens, and is a marker of immune response. We know that in breastfed infants, the SIgA is extremely high, because there it is also influenced by other factors in the breast milk. What we saw in our 5-HMO supplemented formula-fed infants is that they had a higher SIgA levels, which were significantly higher when compared to the control and moving in the direction of breastfed. Another important marker is the gut permeability marker alpha antitrypsin, which the higher it is, the more permeable the gut. So we saw that in the test groups, there was a significant lower amount of alpha antitrypsin marked, which is a marker of a better gut barrier function in the test groups. In conclusion, I showed you that 5-HMO supplemented formula has been clinically shown to shape the gut microbiota and support an overall improved gastrointestinal environment. By shifting several bacterial taxa, including Bifidobacteria, as well as changing the gut ecology towards the one observed in breastfed infants. Also, the test groups had a lower prevalence and abundance of the pathogen associated with diarrhea, enterotoxigenic Clostridioides difficile. We also saw in the test groups an increase in fecal SIgA, which is an important marker of immune response, moving closer to breastfed infants. Lastly, we saw a positive effect on gut health markers by the decrease in a marker of intestinal permeability, alpha antitrypsin, which suggests that HMOs support healthy gut function and maturation. To end, I want to share some take-home messages of my talk. The early life is really a critical window for development, where we see that the gut microbiome and immune system are established and matured. This resilient microbiome really forms the basis for a long-term health by reducing vulnerability to infection and disease. Many factors influence this gut microbiome development in early life, like, for instance, delivery mode, antibiotic use, and infant feeding. And it's there that breast milk really contains all nutrients and bioactive components to drive optimal development of the infant and its microbiome. Especially HMOs, or human milk oligosaccharides, nurture an age-appropriate microbiome. And I showed you data where it was clinically shown that HMOs support gut immune and barrier function, likely linking their effects, likely linking this to their protective effects from infection. I want to thank you very much for your attention,